To today's class. Today, our class is about Golgi complex, or also known as Golgi apparatus. It is a cell organelle named after its discoverer, Camillo Golgi. It was discovered in 1898. Kagel in 1914 named it Golgi complex. Dalton and Felix in 1954 provided its electron microscopic structure. It occurs in all eukaryotic cells except mammalian RBCs, sieve tubes of angiosperms, sperms of bryophytes and pteridophytes. It occurs in two different morphological forms, localized and diffused. Let's try to understand what it means. In localized form, the Golgi complex occurs singly and occupies a fixed position between the nucleus and the secretory pore which is found on the plasma membrane of a cell. They are seen in thyroid cells, exocrine cells of pancreas and mucous cells of intestine. Whereas in the diffused form, several units of Golgi complex are found scattered along with the elements of endoplasmic reticulum. Each unit is called as a dictyosome. It is found more in number in actively secreting cells. They are found in nerve cells, liver cells, plant cells, and also the cells of invertebrates. Let's take a detailed look at the structure of a Golgi complex. As you can see, we can see three different components in a Golgi complex. The flattened structures are called as cisternae, and as you can see, there are spaces between the cisternae referred to as the intercisternal space. There are two sides for the Golgi complex. One is referred to as a concave side, also called as the maturing phase or the trans phase. And the other is the convex side, also called as the forming phase or the cis phase. In the forming phase, transition vesicles are seen. They are drop-like structures as shown in this picture here. They detach from the endoplasmic reticulum, later combine together to form flattened structures called cisternae. The third component is the secretory vesicle, also called as a Golgian vacuole or the secretory granules. It is represented here. They are actually pinched off from the cisternae or the flattened structures, and they later on develop into the primary lysosome containing hydrolytic enzymes in them. Let's try to find out the relation between the endoplasmic reticulum, the Golgi complex, and the lysosome. As we have seen in our earlier class, the endoplasmic reticulum, which are of two types, rough endoplasmic reticulum with ribosomes and smooth endoplasmic reticulum, are actually attached to the nucleus. From the smooth endoplasmic reticulum, drop-like structures called as the transition vesicles are produced, which later on combine to form flattened structures called cisternae. From the cisternae, secretory vesicles are pinched off, which develop into lysosomes or primary lysosomes. Now let's take a detailed look at the functions of Golgi complex. The most important function is associated with secretion. A variety of cells secrete substances like mucus, sweat, tears, saliva, enzymes, hormones, even digestive enzymes and hydrochloric acid, blood proteins, etc. in animals as well as in plant cells. For example, in the case of plant cells, cell wall secretions, gum, wax, etc. are being produced by the Golgi complex. Second is the storage of such secretory products. Third, we can see complex carbohydrates are being synthesized with the help of Golgi complex. Complex carbohydrates in the sense cellulose, pectin, hemicellulose, polymucosaccharides, hyaluronic acid, chondroitin sulfate, etc. Formation of cell plate during the anaphase stage of mitosis is also produced by Golgi complex. Formation of cell wall and formation of acrosome of sperm, which is also a modification and a maturation of the Golgi complex. Formation of yolk, also called as vitellogenesis, that is in the case of animal oocytes. Golgi complex is associated with the formation of yolk. Formation of lysosomes from the Golgi complex is also another important function attributed to the Golgi complex. That's all for today. Thank you and have a nice day.